So this is pretty much like the first slide. Few things that I want to highlight. DNA has to be replicated, you know, cellular components of eukaryotic cell have to be amplified. FTSC protein in bacteria forms the cleavage furrow. Okay. This is mostly about that doubling time. Okay, so know what the generation time is, know what how every generation time, every like 20 minutes, bacterial population doubles. Know that the lab generation time is shorter always because nutrients more abundant. Understand that generation time is different for different species. This is kind of self-explanatory. That's what we talked about yesterday. Remember that task with numbers? Practice it. Okay? So if I tell you we start with 200 cells and in an hour we have 800 cells, what is generation time? And tell me it's 20 minutes. No, sorry, 30 minutes. We start with 200, end up with 800 in an hour, it's 30 minutes. 200 become 400 in 30 minutes and 400 become 800 in another 30 minutes. Is that clear? Okay. So calculate either the final number or the generation time. Growth curve. Phases. What's going on with bacteria in each phase? What about the availability of nutrients? What about the accumulation of metabolites? Okay? Does that make sense? You just follow the guides. What are persisters? Cells with a slower metabolism. When do they appear? Death phase, yes. Decline phase. Understand the principle, the working principle of hemostat. As one student once wrote it down, nutrients in, wastes out. And that's practically it, okay? So I have to understand that it maintains nutrient-rich environment in which, so microbes in a chemostat exist in a balance pretty much between the logarithmic through stationary phase. They do not die, okay? Metabolic waste of fermenting bacteria. What two types of fermentations we have? Acidic fermentation is going to be some sort of lactic acid, and alcoholic, so alcohol is going to be metabolic waste, right? Cell counting methods. So we have discussed various types. I'm not going to list all of them, things like, you know, Coulter counter and turbidimetry. You have to understand which methods let you to count live cells, okay? And recognize the method by description. You saw the questions on the quiz, something of that sort. Is that clear? Okay. Advantages and disadvantages. When I ask this, I mainly focus on live versus not live, and time, bless you, time consumption, and probably most important, the, how we call this person? Whoever does the method, I just blank words. The bias, experiment, the science, the researcher bias, okay? The technician bias. When you count cells yourself, you're biased, okay? So this, you know, serial dilutions, okay? idea of filtration, that it retains the bacterial cells on the filter so you can kind of concentrate them. Most probable number, talked about it. It does not give you the solid number, it gives you the probability. Okay? Measures live cells. So answer those questions. Turbidimetry measures optic density or light absorptions. It's the same idea, right? It's kind of the higher the bacterial density, the more light is being absorbed. No, you cannot. Advantages, disadvantages, figure it out. Non-biased, can be automated, you know. Elements, so okay, elements that form biological macromolecules. That list: carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus. That's it. Know where you're going to find them. Phosphorus, DNA, RNA, proteins, right? Phospholipids also. Roles for the sources of the elements. We talked about it. If you answer that main source of phosphorus is air, it is wrong. This is kind of an easy part because a lot of that stuff is obvious. Phosphorus is going to be mineral deposits. Oxygen is going to be from the air. Roles for the ions: potassium, sodium, chloride, ionic balance, iron, magnesium are enzymatic cofactors. Calcium, enzymatic cofactor. Microelements, enzymatic cofactors. Whether they get them from mineral deposits. Understand the idea of essential nutrient. Anything that microbial cell cannot live without. It can be iron. Iron can be an essential nutrient. Does that make sense? Remember we talked about auxotrophic mutants. Mutants that, for instance, do not have enzymatic system to produce tryptophan. Tryptophan will be essential nutrient for that auxotroph. Got it? Diffusion, simple, facilitated, no energy along the concentration gradient, facilitated, requires transmembrane proteins. Active transport, same stuff. By this time in your life, you have to be able to wake up during the night and recite all the properties of diffusion and active transport seriously. Big particles. Endocytosis, 3 types. Phagocytosis by a phagosome, pinocytosis by a pinosome, receptor-mediated endocytosis by an endosome. Out of the cell, exocytosis. Which one is specific, which one is not? And receptor-mediated endocytosis is specific. Osmosis. Same as with diffusion. You have to be, you wake up in the night and you tell everybody about osmosis. Okay? Regulation of cell shape and rigidity. Cells are usually in hypotonic environment. So due to osmosis, they swell and become turgid, T-U-R-G-I-D. It's a fancy word. It's pretty much like bouncy, okay, inflated. Cell wall prevents them from lysis. Environments, okay, relative term, hypo-iso, hypertonic. Understand swelling, shrinking, not changing. Direction of water movement, right? Why the bacteria do not burst. They have cell wall. How amoeba survives in a hypotonic concentration. It pumps out the water using the water vacuole. High salt concentration. Bacteria maintain high salt concentration inside the cell. Does that make sense? colon. In one phrase, I don't mean, okay, so one phrase, I don't mean that you put all 4 words in one phrase, God forbid. What I'm saying is, when you want to describe mutualism to someone, you tell, mutualistic relationship is when both participants benefit. Commensal, one benefits and one is neither hurt or is benefited, okay? Parasitic, one is hurt, one benefits. Does that make sense? Just practice this. You're going to get a question about these relationships, guaranteed. Non-symbiotic, same, we discussed too, you have to know them. Okay, difference between symbiotic, non-symbiotic, and not obligatory, not mandatory. Planktonic form biofilm, all this stuff about biofilm. Planktonic is suspension biofilm on the surface. Understand the roles of extrapolymeric substances. It's extracellular matrix that increases the adhesion and it protects microbes from desiccation. Yeah, it should be here. Good that we found. What is its role? Okay, so EPS. What is the role in the biofilm formation? Primary, secondary colonizers, order of colonization. Okay, how would you characterize relationships between cells in the biofilm? It's either mutualistic or commensal, or both. Okay. Critical density, just too dense. At this moment, microbes start to produce quorum sensing molecules. Okay, so quorum sensing molecules for gram negatives, homoserine lactone, for gram positives, short peptides. Okay, just know these 2 types. Why they are different? We actually hypothesized about it in the classroom. Different structures of the cell wall facilitate different transport mechanisms. Okay, what is the selective pressure to evolve specifically to use homoserine lactone versus peptides? We don't know, but it turns out sensing molecules are distinct. What is auto-induction? What are the auto-inducers? Pretty much it's positive feedback loop. These molecules induce their own production. Let's go over that page so I know where to start tomorrow. Saprobes, bacteria and fungi, thrive on dead organic matter. Feeding mechanism, remember exoenzymes that destroy the substrate outside and then nutrients diffuse into the cell. Parasites, and the difference from saprobes, live on the living organic matter. Ectoendoparasite, that's simple. Obligate parasite must be in parasitic relationship. This, remember that 5 types of microbes based on the oxygen requirements? You have to know them. Questions like description. Microbe must have oxygen. Microbe must not have oxygen. Microbe grows on top of the tube, on the bottom of the tube, in a very narrow range in the tube, all over the tube without any difference. You have to be able to recognize this stuff. Does that make sense? Goldilocks, because microaerophiles want really precise concentration of oxygen, no more, no less. Am I clear? Okay, before we move on,